Imagine having to assemble a dictionary of the English language, but every time you open a new book, you find words that you've never seen before. Many of the words are unique, found only in that book and nowhere else. Likewise, in genetics, hundreds of thousands of new genes are being discovered by DNA sequencing, and their beginnings are a major puzzle. Coming up on today's edition of Origins, Orphan Genes Puzzle with Dr. Paul Nelson. Hello and welcome to Origins. I'm Ray Heiple. It's an honor to be your host today. During this program, we showcase interesting guests who present evidence from science along with other important facts validating the truth of creation and the accuracy of the Bible. Our guest today, Dr. Paul Nelson, studied evolutionary theory and the philosophy of science at the University of Chicago. He's currently a senior fellow of the Discovery Institute and adjunct professor in science and religion at Biola University. Paul has been involved in the intelligent design debate internationally for more than 2 decades. And we're happy to have you with us, Paul.
Hey, it's great to be here.
So we're going to be looking at orphan genes. I know what orphans are, but what are orphan genes?
Well, orphan genes are sequences of DNA that code for protein, but they have no apparent relatives. That is, when you align them, you can't find any matches in the genetic dictionaries of life, and they create a real puzzle for evolution. But before we get there, let me give you a little thought experiment. Let's suppose we are sitting in a Starbucks here in Pittsburgh, and I say to you, I can walk to New Kensington in 1 day. And that's about 20 miles. And you say, yeah, I know what that would require. You can do that. That's possible. All right, same Starbucks a week later, and I tell you, you know what? I walked to Buffalo. Now that's 215 miles. And you say, okay, but never in 1 day.
Or in wintertime.
Or in wintertime. You're going to need shelter. You're going to need a change of socks, at least probably more than one. It's possible, not in 1 day, only with help. Okay. All right, same Starbucks. Another week later, I tell you, I walked to Honolulu. Okay, now you might, grab your cell phone and say, no, I know a good psychiatrist. Okay. There's no way, no matter how much time you have, it's over 4,600 miles, and there's this little body of water on the west coast, on the west coast of the United States. All right, so let's apply this to biology.
All
right. With a question like, can you walk to New Kensington versus can you walk to Honolulu, we have ways of answering that, all right? What do? Well, we say, what do you need to walk, right? What does the mechanism of human walking require? So we have some knowledge of the process itself, but we also have accurate maps of the terrain. So we can assess my claims about walking to New Kensington versus Honolulu with respect to everything we know about the world. We can do the same thing with biology. What's possible versus impossible and why? All right? So what do organisms require just for their normal function and reproductive capability? And we have maps, so to speak, of where organisms are located with respect to each other in terms of their genetics, development, anatomy, and so forth. So this is the same kind of information we can use, as in the walking case, to evaluate claims, particularly about evolution.
Knowing our abilities and knowing the terrain. Same thing with an organism or walking.
Right, same thing.
Right. I'm with you so far.
Let's take an example real close to home, our relationship to chimps, all right? Now, here is a protein that's circulating in your bloodstream right now. It's an oxygen carrier. This particular protein, if you look at it in a human, here's the sequence. Now, these single letters represent amino acids, so this is the amino acid sequence to build that protein in humans.
That's how many amino acids in just that one protein?
In that delta hemoglobin, right? It's a complicated molecule. Here's the same sequence in a chimp. They're identical except for one difference right here. We have a methionine there, they have a valine there. Otherwise, these two proteins are exactly the same between chimps and humans. But you know, there are many proteins in chimps and humans that are exactly the same. Here's a partial list. We could fill this board many times, which raises an interesting question. If down at the nuts and bolts level we're so similar in terms of our genes and proteins, why don't we look like this? Because we don't, right? I mean, I know some rather hairy people, but they don't look like that. If you're lucky enough, you look like this, right? I'm not. But let's do a little comparison here between our two biologies. If you look just at our anatomy, there are enormous and very interesting differences between chimps and humans. For instance, we have a chin, they don't. They have no eyebrows, we do. We have an opposable thumb, lets us do this. We have great dexterity, they don't. And on and on. In fact, some anatomists have said you can distinguish a bone in a chimp from any bone in a human just by visual inspection.
Every bone's a little different.
Every bone's a little different, and some are very different. You run through this list and you see there are many, many behavioral differences between our two species. The puzzle is, how can that be if we are genetically nearly identical to chimps? This disconnect has been a real puzzle in evolutionary theory for several decades. In fact, I remember as a student reading this paper. It was published in Science in 1975 by two researchers at Berkeley University. What they pointed out is if you look at the genes and proteins of chimps and humans, in many cases they're identical or nearly identical, yet the biology of the two species in terms of their behavior, their anatomy, and so forth is quite different. And they said evolution must be occurring at two levels, because down at the nuts and bolts we look like we're largely the same. You rise up and look at the differences, we're very strikingly disparate. Now, there's a cynical view that you can take of this. Jonathan Marks at the University of North Carolina says, look, if the overall biology of the animals tells you they're very different and the genetics tells you that they're nearly identical, it follows that the genetics is telling you something relatively trivial about the overall biology. So he says, look, if you look at the genes and proteins and it says you should be in the same species, they're telling you the wrong thing. I think he's right in a certain level, but I'm not going to take the cynical view. I want to take a different view and look at what evolution is as a theory. Evolution is not primarily a theory of similarity. Many people think it is, but really, similarity just gives you more of the same thing. Evolution fundamentally is a theory of transformation. What it says is it's possible to go from species A to species B by some natural pathway where you have major changes occurring. You're transforming that species. So, when we look at Darwin himself, Darwin actually only used the word evolution once to refer to his own theory. He had a different name. It's longer, but in many respects, it's more accurate. He called his theory descent with modification. The reason I like this name is it captures both elements of what any evolutionary theory would need to explain. Think about it this way. You've got descent, that's where you get your similarity. But that's not enough because, as I said, similarity just gives you more of the same thing. What you need is modification. You've got to transform that species over time, and the joint or the conjunction of these two is what really gives you evolution.
So the descent is hereditary descent. From one generation to the next, we use the word descent for that.
Right.
But over time, there's got to be this transformation.
There's got to be transformation or it's not evolution.
At the same time, right?
At the same time. Both things have to be happening. Now, this is important because Darwin himself knew that if this failed, if you didn't have a mechanism to change living things, you wouldn't have evolution as he understood it. In fact, he proposed a test in The Origin of Species saying some of these transformations might be biologically impossible. Here's how he put it. He said, if any feature of a living thing could be demonstrated that it could not have arisen by numerous successive slight modifications, my theory would absolutely break down.
Does he mean that his whole theory or just for that particular modification?
Well, it really applies to the whole theory. Because what you would say is, here's a feature of a living thing that really exists. And Darwin said all living things are connected. So if some living thing is isolated from the rest of life, where did it come from? So it's very important for Darwin that everything that we see about organisms can be fit into one great tree of life. So this was and continues to be a very serious test for evolution.
It seems like if even one thing could be found that you couldn't explain, then it would affect everything else.
That's right. It would be a significant challenge because this aspect of evolution would fail. And descent alone doesn't give you evolution as Darwin understood it. It's often said that evolution, in Darwin's sense of a single tree of life, is an established fact, right? You just don't doubt it. Actually, in 2009, New Scientist, a major science journal in the United Kingdom, had a cover story, "Darwin was wrong." Now that got a lot of attention, and many evolutionary biologists were very upset. They said, "We'll never have anything to do with this journal again. They're just pandering to creationists." But Graham Lawton, the science writer who wrote the piece, defended himself online. He said, "Look, I'm just reporting on what's happening within evolutionary theory itself. Many senior evolutionary biologists are bailing out of Darwin's single tree picture. You can kind of see it here in this nice illustration. You've got a root, common ancestor to all of life, and all the rest of life is going to spring from that root." These biologists who are uprooting Darwin's tree, as the article puts it, have said, you know what, that's not true anymore. So if we want, we can go into a little more detail on this and talk about why orphan genes and similar puzzles have caused Darwin's tree to be uprooted.
Before we do, Paul, we have to take a break. You're going to want to stay with us. Modern evolutionary theory says Charles Darwin was right, and yet we have this magazine that says he was wrong. Find out the solution to this when we come back. Welcome back to Origins. We're talking to Dr. Paul Nelson, who's been sharing some fascinating things about orphan genes. Paul, we left off looking at this cover story of New Scientist magazine saying that Darwin was wrong.
Shocked a lot of people.
I imagine it shocked me to see that.
And the funny thing is, when you look at the backstory to the story, so to speak, the science writer Graham Lawton says, look, I was just reporting on what's going on within evolutionary theory itself. For instance, the work of this man, Carl Woese. He's dead now, he died a few years ago, but he was a molecular biologist at the University of Illinois and very prominent member of the National Academy of Sciences. And towards the end of his career, he was quite outspoken to say biology has to go beyond what he called the doctrine of common descent. And that capital D there, that's not a typo. He used that word in a sense of something standing in the way of scientific understanding.
Was he referring to Darwin's theory when he said those words?
He was referring to that single tree picture where all of life springs from what's called the last universal common ancestor, sort of your ultimate grandparent and mine. So if we look at our lineages, probably you have Northern European ancestry, I do too. We would converge to kin in common, remarkably recent in human history. So if you did your genealogy and I did mine, we would find that we have relatives in common somewhere in Northern Europe within just a few hundred years. In fact, for any person in the audience, we can start tracing those lineages back and you'll discover you're related to every other human being on this planet. Remarkably recent in human history.
Would evolutionists agree with that?
They sure would, but they would say not only are you related to every human being, you're related to chimps and your dog and in fact the bacteria in your gut. Okay. Now, what Woese was saying is that single tree picture has broken apart at the bottom. The so-called last universal common ancestor, or LUCA, as it's often abbreviated, never existed. And there must have been multiple trees that came to be independently of each other early in the history of life. Now, what kind of evidence is leading him to this sort of position? Well, let's look at it. One thing that we didn't know, that nobody knew when I was a student, was just how diverse genetics is on planet Earth. In the mid-90s, automated DNA sequencing, this is a somewhat older generation DNA sequencer, didn't exist.
Imagine they're smaller today.
They're smaller. In fact, within our lifetimes, high school biology classes will have DNA sequencers about this big.
Wow.
And they will, for a semester project, sequence the DNA of a species no one's ever looked at before. So the technology has become so inexpensive and so rapid that, this is not an exaggeration, it will be within the grasp of a high school AP biology class. So this is an old machine, I need to update the slide, but the point is, this technology didn't exist when evolutionary theory was formulated. And what it has shown us is something really astonishing. Now this curve should blow you away, okay?
When you explain it to me, maybe it will.
All right. Actually, you can see it only goes to 2008. Now this line goes up and off the screen. What this represents is the DNA information stored in GenBank. GenBank you can think of, well, it is literally a computer database, but you can think of it as the dictionary of genes, very much like a dictionary of words. So if I gave you the task of writing an English dictionary, you would have a curve that would be initially similar to this, where you'd get a lot of new words that you'd never seen before, but eventually your curve would flatten out. And you'd come back to me after, let's say, a decade, and you would say, look, I have got and assembled a comprehensive dictionary of the English language, right? There just aren't any more new words to find. And even the new phrases that I find, like hip-hop, right? Hip-hop is a conjunction of two words that were previously present in English, or rip-off. Right? Mm-hmm. I'm old enough to remember when rip-off entered the English language because hippies in the 60s needed a word to describe how they felt when someone sold them a bag of oregano, right, instead of weed, okay, to be blunt about it. So, man, he ripped me off. Well, rip and off had an existence in English prior to, all right, so your dictionary is going to, be complete. What's happening with genes, on the other hand, is this curve is going up out of sight. What that means is that the genetic words, if you think of it that way, in biology is vastly greater. The number is vastly greater than anyone realized.
So they're finding brand new words, so to speak.
All the time, all the time. So this is what I just said. Before the mid-1990s and that DNA sequencing technology, we had no idea. We had no idea just how diverse things are. And one of the phenomena, if you will, that's causing this dictionary to grow so rapidly is what are known as orphan genes. These are genes that are found in a single species. You look in another species, they're not there. In fact, you can take these genes and compare them to the whole of life, and you don't find any matches. That's why they're called orphans, because their putative evolutionary parents can't be found. Now, let me show you how this is discovered. A little cartoon here. Here's a sequence of DNA that we've just determined, right? Took it from a new species we're looking at, and long strings of A's, T's, C's, and G's, long enough to code for a protein. We determine our sequence, and then we submit it to GenBank. So here's our genetic dictionary, and just as if I gave you an English word you'd never seen before, you'd go to your Funkin Wagnalls or your Oxford English Dictionary and look for the match. In this case, we get a match. So we align these sequences base pair by base pair. This one codes for a ribosomal protein, so does that one. Now, this one can join the dictionary too. With orphans, that doesn't happen. We look for the matches and they're just not there. This clearly codes for protein because it has the features of a gene that codes for protein. But when we put it in the dictionary, no match.
So even with the human genome being totally completed, right? And they have mapped all of our DNA and throughout all of our cells, would they still find something that wouldn't already be there?
You still find orphan genes. In fact, orphan genes are present in every species that's been looked at so far. In some cases, they're a very large percentage of the DNA of that organism.
And they're new within that generation, like you don't see them in the previous generation.
Well, they would be present any time that you could take a sample. But remember, when we're looking at genes, we're looking at just the organisms present today. So we can only infer about the past. But as you look at the total number of orphans, they vastly exceed the shared genes. So some genes are shared between species, but the orphans in total number greatly exceed that. In fact, as these authors say in Germany, the number is much greater than that for known gene families. And we find them everywhere, in enormous numbers. So this little creature, the water flea, Daphnia, had its genome sequenced recently. And 36% of its genes show no homology, that is, no similarity to genes in GenBank or other databases. Over a third of its genes are found just in this creature.
Just in that creature.
Or look at this one, this tick. And by the way, on a quiet afternoon in heaven, I'm going to ask God what he had in mind with ticks, okay? Really don't like them, all right? Look at the number here. 71% of its genes have no matches to in other databases. And these genes aren't just peripheral players. All right, salamanders, many species can regenerate their limbs. It's a remarkable feature, right? Mm-hmm. Limb regeneration in salamanders is apparently mediated by an orphan gene.
Does that mean they don't know what gene does it?
No, they know what gene does it, but when they compare that gene to other amphibians or to other animals, they don't find it elsewhere. So this key feature of this species is found just in that species, and the gene that does it, and its protein product, appears to be just in these guys. Or, another example, freshwater polyp hydra, which some people remember from high school biology, this critter here, its very form, its anatomy, is specified by a species-specific orphan sequence. So, bottom line is, these genes are present all throughout life, and there's a problem for Darwin's picture. If we start with the last universal common ancestor, we can define a radius that extends from that point. And this line is set by what do organisms need, how much mutations are possible, and how much time is available. That gives us a sphere within which, if evolution is true, we should find all known genes and protein products. But this space is only a tiny fraction of what's available to an intelligent designer. All right, put it down there. A designer can go anywhere he wants in this larger space. He's not bound by that radius, so to speak. So we can let the data settle it. If we map a new species, look at its DNA, is it going to be in here, or will it be located out here? So I'm very, very excited about this because it's a place where the predictions of intelligent design and the predictions of Darwinian theory radically diverge.
What do evolutionists say about the orphan gene?
It's a major puzzle. It's a major puzzle, currently unsolved, and it's something I get very enthusiastic about because, frankly, I think it's a place where new discoveries in science are lending great support to an intelligent design picture of life. Now, there are more details to work out, but I think it's tremendously exciting.
Well, where do we go from here? After you've mapped the genes of different organisms, what do they do with the orphan genes? Do they just go into the bank separately?
They go into the data bank, and in fact, I'm involved with a research project right now called Orphan Base, where we are trying to get a comprehensive look at all these sequences and what they might mean for biology.
Do you ever find where there was an actual likeness to another orphan?
Oh, sure. Sometimes orphans turn out to have relatives. Then they can no longer be classified as orphans. But the total number of orphans known continues to rise in a dramatic way.
So it's not catching up?
No. We're not catching up. The dictionary of life gets bigger and bigger and bigger every time a new sequence or a new gene or a new species is looked at.
You know, I teach apologetics at a Christian high school, and one of the things that I like to try to impress upon the kids that apologetics and evangelism are not the same things.
That's right.
Evangelism is preaching the gospel, witnessing to Christ, but apologetics is defending the truth claims of Scripture and defending the truth of God in the world as we see it, as we perceive it. We've almost given up on the world. What do you think has happened?
I think the problem is that many Christians are intimidated by science. And this wasn't always the case. If you look back at the history of science prior to Darwin, much of the very best work was done by Christians. But with the middle of the 19th century, the philosophy of materialism or naturalism comes to dominate scientific thinking. And we still live in that intellectual world, a world in which it's said science can only use natural laws and chance processes to explain what we see. So if science, for many people, has that character, they feel like, well, that belongs to some other philosophy. I will draw my understanding of God from His Word and from my church fellowship, and I'll let this other worldview have the natural world. I think that's a terrible surrender. That's a terrible cost to pay. And Christians need to have the confidence and really eagerness to go back to the natural world and reclaim it for a theistic worldview. If this world was created by a mind like mine, but vastly more powerful, I have some hope when I go out and do the hard work of research that I'm going to discover what that mind did.
Well, that's been fascinating stuff. I really appreciate you coming with us, Paul. And I appreciate you joining us as well. We've seen some compelling evidence that random mutations simply are not able to account for the complexity of life, even at the genetic level, that there are these orphan genes out there that evolutionists really don't know what to do with. But when we look at the scriptures and we see there's an intelligent designer, then it confirms something that we like to say on this program, and that is we know what the Bible says is true, and the proof is all around you. Thanks for joining us for this edition of Origins. We'll see you next time.
Thank you for watching this edition of Origins. For a DVD of this series, you can order online or send a $12 donation to cover shipping and handling. And write to Origins Program, number 1809, Cornerstone Network, Wall, Pennsylvania, 15148.
This presentation was made possible by the faithful prayers and financial support of you, our Cornerstone family.